have a pretty diverse audience. We have some biostat students, some French students, some bio students, and some others. And I wanted to introduce our speaker, Jesse Boehm, who's coming to us today from the Broad Institute in Cambridge. It's affiliated with MIT and Harvard, and Jesse went to MIT for his undergrad. He got his PhD at Harvard.
Wait.
And he has his HS diploma from Concord, Carlisle. So we're very, very proud to have him here. So, this is probably his first time back, and certainly he thinks that this is a kind of an improvement over the old building.
This is amazing. Absolutely unbelievable.
Yeah, so I'm going to let him take it from here.
You know, to actually be invited back and to come to your high school is, I think, like the pinnacle of your professional success. So this is like the most exciting lecture I've ever given. So I was thinking, when Sandy said I could present in the auditorium, I was thinking about how terrifying it would be, and then I was thinking about AP Calculus with Mr. Peter Atlas. With Peter Atlas, that was equally terrifying. But actually, I think this is, to come and speak to people really on the early stage of their careers, it's hard to actually know exactly how to communicate what an exciting moment in science this is. And I think through this early example of the Broad collaborating with Sandy's class to test this notion of crowd science and problem solving, we've gotten very excited about engaging high schoolers and the general public in many ways. And so I wanted to come. I can't speak French and biology and biostatistics all at once, but I wanted to come and try to describe just a little bit of the reasons why I and my colleagues are just so fundamentally excited about this moment in time, 2015 biomedicine, especially as it pertains to cancer. Take you a little bit through what's actually happening now. I always felt it was difficult reading the New York Times or going to biology class. It's very hard to actually know what's at the very cutting edge, and I was hoping to give you some of the sense of what's the most exciting things going on today, and then if we have some time and answer questions. But hopefully this could be really interactive. I certainly don't want to make this boring. I remember sitting through, all sorts of auditorium things with some lecturer talking about all sorts of boring things. I'd rather this be exciting. So that's why I titled it Curing Cancer. We could give a biology update about cancer and cancer cells and what they do and how they divide. But I think at this moment where patients are going to the clinic, seeing their doctor, using the genetic material of their particular tumor to help inform what drugs to get, this is an unbelievable moment in cancer biology. And it's making us and others feel really excited that we're on the cusp of real cancer cures. And I'll tell you why in a moment. So 10 years ago, a title that's called Curing Cancer, people would say, it's just crazy talk. Today, it's actually a reality. We think in the next 20 years or so, it'll be very likely at the current pace that cancer will be a disease that people will get, but you'll be able to keep it in check. Many types of cancers, you'll be able to keep it in check with a special combination of drugs that's tailored to you and your genetic material. This is all made possible post-Human Genome Project. So where has cancer medicine been? Where are we today? And how did my graduate years in CCHS and all this map on it? So when I graduated CCHS in Peter Atlas's class in 1997, we were still treating cancer. You had breast cancer. You got some random drugs that scientists had discovered through sort of empiric random testing over the years. And for some people that worked, for other people that didn't work. But we still treated cancer by the part of your body that it came from. As we move forward, and I'll talk more about post-Human Genome Project, we've started treating people's cancers based on which gene is actually changed or mutated. Can I use mutated? Okay. I'll assume no prior knowledge, but maybe some. Which gene is mutated in that tumor, and the patients with that gene could get a particular drug that's known to be effective for their type of tumor. And that's called precision medicine, or precise cancer medicine. In the early 2010s, that's beginning to become a reality. So 2015, here's where we are today. We're starting to see precision medicine really take off in major cancer hospitals. And where we're going is a future in which the DNA from your tumor, hopefully not your tumor, the tumor that ends up being sequenced in the clinic, one can customize a therapy just for you. So I'll describe how we're trying to get there. But in 1997, I graduated CCHS, and 2001 from MIT, we were still all the way at the other end of the spectrum. So just imagine when you'll go to some higher education, you'll do something. Just imagine what 20 years from now is going to look like. And I think that's where you want to be. It's impossible to imagine, right? You couldn't even imagine iPhones or anything else 10 or 15 years ago. So thinking about what's possible in 20 years is a difficult activity, but an important one as you think about the future. So what does this look like when it's successful? It looks like totally remarkable progress. On the left, this is someone with a melanoma, a disease of pigment-producing skin cells that's metastasized all over their body. This is one of the early success stories where scientists figure out what gene was actually causing this melanoma, and drug companies knew how to build a drug that blocked this gene, and if you gave the right drug to the right patient, the tumors would just completely disappear. This is only possible if you know the gene, you know the drug, and you can give it to the right patient. So this is totally extraordinary, at least as far as I can tell. And this is now being replicated. And it's actually this biostatistics course that's trying to show how scientists are making this possible for other types of genes and drugs. I won't talk more about this today, but it's important to recognize there's a really exciting new front in cancer biology just in the last maybe 18 months that's taking the popular press by storm, taking the scientific community by storm. And this is the notion, an idea maybe 30 or 40 years ago that was never fully realized, to use the body's own immune system and teach it how to fight the tumors that are in your body. If you remember, a tumor, and I'll talk more about it, is just a group of your cells that is a little bit different from the rest of your cells in the body. And your immune system is designed to go around and look for things in your body that don't belong, usually viruses and bacteria. So why can cancers grow when you have a normal immune system? It turns out that tumors turn off the immune system. They sort of enable it to break itself, break and turn off. And some of these new drugs, which have been featured in the New York Times and all sorts of popular press, are now teaching the immune cells how to turn themselves back on and find the tumor cells that they couldn't see before. This is leading to not just initial successes, but long-term successes. Patients with this late-stage melanoma can now survive 60% of them over 18 months, where just 3 or 4 years ago that would have been under 5%. So totally extraordinary. It's totally unclear exactly all of the details, how it's working, but this is an example of a front in biomedicine that you in your future graduate work or whatever could help to figure out how to uncover. Not only is science changing, but one of the things I'm most excited about and the Broad is most excited about is the culture of science is changing. Science, when I don't know if anyone remember Mr. Kazuski, is he still around? My high school biology teacher. In my science class, I learned a lot about cells, how they worked, and DNA, and there was a lot of facts and understanding, and it felt like science was something really specialized, that I had to spend 10 or 20 years and work in a lab and be in a basement somewhere and get these graduate degrees. And in the past, that's true. It has really been only a set, you have to really overcome certain barriers to be part of the scientific community. But today, and I'll show you some examples, patients often have the answer to curing disease within the genomes of their bodies and are volunteering to be part of the scientific process, which has always happened through clinical trials and testing, but through social media and advocacy groups, people are saying, I want to be part of the solution. This was largely the way that HIV began to be managed and treated. That patients went in the streets and said, I want to be part of the solution. And this is now beginning to happen in cancer and other diseases. And on this sort of smorgasbord of careers, I'm trying to show that there's many ways of being involved in science, from law and marketing and communication, policyholders, voting, working at a company, working as a doctor, and all of us have a role to play in the scientific process. That's more true today than it's ever been in the past. As just one example, we're experimenting at the Broad with reaching out to cancer patients directly and inviting through social media, through Twitter and Instagram and Pinterest and all these things that I'm just learning. I'm like 5 years behind, but you're all over all the latest things. But the first thing patients often do when they receive a diagnosis of cancer is they go online, they Google it, they look around, and they build social communities. And they often want to be part of research teams that solve problems. It turns out that the majority of cancer patients, and this is true in other diseases as well, are treated at a local hospital near their house. That's actually great for the patient. That helps them receive care. But if you can think about it, there's thousands of hospitals all across the country, and it's sometimes unclear how, if you're being treated in, some hospital in the middle of, Midwest somewhere. Everyone always picks on the Midwest. Middle of Midwest somewhere. how do you actually be part of the major efforts that happening in some of these major hospitals? But social media provides a way to do that, and so we're going out to patients and directly inviting them to be part of the research. You don't need to be a scientist to respond to a Twitter campaign. Where does the Broad stand in all of this? So, there's many places to work on cancer. You can work in a lab, in any hospital, in any institution, anywhere across the country. What we've started to realize at this really amazing moment in biomedicine, and at this really amazing moment where the culture of teams and team science is changing, there's a role to play for a new type of institution. The Broad is a building, a set of buildings in that orange dot. This is Cambridge on the left and Boston on the right. But it's really a horizontal connector, kind of a community. Harvard and MIT, I don't think ever really collaborated on anything substantial before the Broad Institute. Is anyone from Harvard or MIT? Before the Broad Institute, they decided they both had different strengths, and they wanted to work together beyond what the two institutions could do separately. The Harvard hospitals, MGH, and Harvard Medical School, and Dana-Farber, all these institutions were doing things separately, but we decided to try to build a different type of institution that acts as an umbrella and brings people together so that, it's actually not competitive to work with someone in a different building, you can actually collaborate. Within this horizontal community all across the Boston area that's become this Broad Institute are a set of 200 labs. And these 200 labs include 30 labs that desperately want to work on cancer, and they want to do this together. But they want to do it in a different way that isn't just repeating what every institute has across the country. We want to have the innovation that tends to occur in academia. If you go to MIT or something, you have really great ideas sometimes. But drug companies know how to solve big problems and scale it up. We try to do those two things together. We try to work as part of teams in a collaborative approach, and we try to do things that will have a big impact on the world, not just for us. So that's kind of different from other institutions. So that's my preamble. That's just where I'm going to, that's where cancer is today. We're at the precipice of something extraordinary. We're at the precipice of something extraordinary from a community perspective. I wanted to now unpack some of those ideas and talk through a little bit of the details about what cancer is, where it comes from, how genes are involved. Some of these things you may already know. You can Sandy, you can fast forward me if I'm going too slow. But maybe just before I stop there, and before I continue, any questions about the overall directions of biomedicine today, cancer, how this relates to being a junior, senior, sophomores, freshman at CCHS? Because I remember being in this role, and no one ever communicated to me where the field was and where it was going, and invited me to be part of it. And I want you guys to recognize that you're about to embark on this careers of your choosing, and in many different career paths, you will want to be part of a larger scientific community. And the earlier you can embrace that, I think the more exciting and fulfilled your careers will be. Too lofty, too ambitious? I'm very excited. Okay, so I showed you this thing before. This patient responded. It was extraordinary. So how did this actually happen? Okay, so in 2002, the earliest days of genetic analysis started to look at tumors like this person's tumor. And they found this one, of the 20,000 genes in the genome, they found this one particular gene called BRAF, that was disrupted in a lot of melanoma patients. There could be any different combinations of genes to give you melanoma, but it turned out that this one gene actually was always turned on in about 50% of melanomas. Drug companies jumped right on this. They said, oh, we know how to block that particular type of protein. It's an enzyme. People with chemistry, enzymes, it was a certain type of enzyme. They could figure out how to block it. And once they built a drug, they knew which patients should get the drug. It was only patients that had that genetic makeup. So instead of launching, sometimes when you start these clinical trials, you have to enroll thousands and thousands of patients to see a small number of people that respond. And that's very expensive and slow. But with these new drugs for cancer that are specific for genes, you only have to launch a smaller trial because you know exactly who's likely to respond to your drug. So the clinical trial that was done was smaller, faster, cheaper, and just 7 years after the discovery, there was already a drug in patients and it was approved This has now been shortened to 5 or 6 years. There was a gene discovery just 5 or 6 years ago that's now already a drug in patients. And so the window of the period of time from discovery to clinical impact is really short. So let's talk about some of the fundamental elements you need to understand anything I said. I'll go through this quickly. You've probably had biology class and learned a little bit about these elements. What is cancer? Well, you saw in this person's body, they were cells growing all over this person's body. It's clearly an uncontrolled growth of cells. You have cells in your body, of course, of many different types. But those cell types divide and stop dividing and grow and shrink and die according to normal cues. Cancer acquires a set of genetic changes which allows those unlucky cells to continue to divide, and they stop listening to the body that's telling them to stop. And so in the case of skin, you might have different types of cells on your skin, and as they acquire these gene changes, they start dividing over and over again, and they can't stop. They can escape from the immune system, is one example. So that's at the cell level, what's happening with cancer. Cancer is also, as you probably know, it's a family of different diseases. We call cancer cancer, but there are many types of cancer. Scientists and doctors use all of these different names to describe different types of cancer, but they really are just naming the part of the body that the cancer comes from. The very most common types of cancers for both men and women, breast cancer, lung cancer, prostate cancer, colon cancer, those tend to be in this category that are called carcinomas. The names aren't that important, just the general concept is cells that protect the outside or even the inside of your body. You have obviously parts of the inside of your body where you need to protect the fluid or the blood vessel or your digestive system from the surrounding tissue. And so these covering cells, either on the inside or the outside, can divide out of control, and those are called carcinomas. There's other types of cancers, less common, called sarcomas. These are from cells in fat and bone and muscle that divide. And you may have heard about leukemias and lymphomas. Leukemias tend to be common in children, although also in adults. These are your blood cells growing out of control. So even though there's all these different names and they're all crazy and they're hard to remember, they're all just naming different parts of the body that can divide out of, so where the cells can divide out of control and go on to form cancer. So in addition to these cells dividing out of control from many different parts of your body, the key element to if you remember nothing else, if you go home and in French class, I went to the auditorium, this 97 CCHS grad was spouting this cancer nonsense. Just remember that cancer is caused by mutations. DNA changes in your cells. Every one of your cells has billions of letters of DNA, and small changes can make those cells grow out of control. These can be little letter changes that turn a gene on or turn it off. They can be big parts of the genome that have too many copies or too few copies. And all of the different things that we think about that cause cancer, actually answering my question, what they have in common is they all produce these mutations in the DNA of your cell. So you probably heard that tobacco smoke causes cancer. It irritates the lining of your airway and produces these mutations. Asbestos, certain viruses and bacteria, I'll talk more about human papillomavirus, radiation from the sun tanning beds, all those things. All of these things cause genetic changes in your cells. And so everything that we know that causes cancer, they're not different things. They all come together in causing these DNA mutations in your cells. Once you know about all of these things that can cause cancer, it's a lot easier to prevent cancer than it is to treat cancer once you've got it. So if you look at the many different causes of cancer, it's actually astonishing that a large fraction of cancers are preventable if we would just, follow the advice that the medical establishment has been telling us. 33% of all cancers are caused by tobacco use. It's like totally incredible. Totally incredible in terms of the impact on individuals' lives, their families, on the healthcare system. If everyone just stops smoking, a third of cancers will be gone tomorrow. We already know this because as tobacco use has gone down, we're already seeing decreases in lung cancer rates. So no smoking. In terms of cancer-causing viruses or bacteria, like human papillomavirus, which actually turns out to cause nearly all cervical cancers, the sexually transmitted disease HPV, you probably know about this new vaccine that's been developed that teaches the body how to prevent HPV infection. And this is actually, in your lifetime, or the lifetime of your children, there will probably be almost no cervical cancer anymore. Zero, almost zero. That's because of this vaccine that if you get before, obviously, you get infected with HPV, your body then can't become infected with HPV, and a large number of sexually transmitted viruses that cause cancer, like cervical cancer, will be prevented. So, we could cure most lung cancer, a lot of lung cancers by stopping smoking. We could take these vaccines and end a few cancer types. That'd be pretty exciting. And that's the result of understanding which things actually cause cancer. So the other important thing is, in addition to these environmental stresses, smoking and sun tanning without sunscreen and all of that, you can be predisposed to getting cancer by the mutations you inherit from your parents. You've probably all heard of cancers can be passed through families. Most cancers are not hereditary. Most cancers are unlucky or spontaneous because of these environmental mutations, but some cancers are hereditary. And so a hereditary cancer means that the mother or father has already in all of the cells in their body one copy of the one or more mutant genes, and some of their offspring, depending on the pattern of genetics, as you've probably learned, can actually be predisposed to getting cancer. You probably need 3 or 4 different genetic mutations to have a full-blown cancer, but if you inherit 1 or 2, you're already on the way to getting there. And to learn which genes cause cancer, up until 2001, for the last 40 years, people just studied these families. They looked to see the pattern of inheritance within the family, and then they tried to use fancy techniques to figure out which gene was involved, and they figured out all of these genes that cause cancer, like the BRCA1 and 2 genes that you may have heard about through Angela Jolie and everything else that are important in breast cancer. So that's the history. That's where cancer, that's where we've been, that's what we've known about. Everything changed in 2001. Does anyone know what happened in 2001? I graduated MIT. Human Genome Project. That's cheating for the teacher. So in 2001, there was this international race. All the details aren't important, but there's a sequencing of the human genome. So in all the cells in your body, 3 billion letters of DNA, and we didn't have the map of what were all the genes that made us. And after the sequencing of the human genome, we had this first roadmap of the world. In this case, it was the roadmap of cells in our body. And this allowed us to now systematically not look at all of the cells in your body. So all of the cells in your body have the same genetic makeup. But a cancer cell, remember, it might acquire 1 or more random changes, random differences. So it would be different than every other cell in your body at 1 or 2 or more specific places. So once you had the roadmap of all the genetic changes in your body, you could analyze the cancer cells and compare them to the normal cells and systematically look for all of the differences. And this is what we do and others. This is where the Broad Institute came from. This is the public part of the Human Genome Project became the Broad Institute, and now our goal is to use those same approaches to cure diseases and not just study them. So if you want to understand, if one person has cancer, that's that nice looking man on the left. If you want to understand the genetic changes that are causing his tumor, you have to do 2 things. You have to map his genome. You take a blood sample and map all of the genetic changes in all of the cells in his body, and your blood's a good place to find a sample of all of your cells. That's called the germline. And then you would look at the tumor cells and look for what's called somatic changes. Those are the changes that occur after you're born in those cancer cells. And by comparing those 2, in any cancer patient now, you can figure out which are the mutations that cause their tumor. You could never do this before, and we started doing this soon after the Genome Project, and we've now done this tens of thousands of times. But since every cancer is actually a little bit different, and I told you melanoma, there was one gene that caused a lot of cancers, but actually this isn't always the case. It turns out that if you look at Sally's cancer and Joe's cancer and someone else's cancer, they'll share some things, but they'll have many differences. And so actually having all this information can be daunting, because now there's all of these genetic changes. If you look at a melanoma cell that's been bombarded bombarded by the sun, there might be 1,000 different genetic mutations, and only 5 of them might be the causative ones. So you have to look across a big population and use statistical tools that you guys have learned about in the front row, biostatistics class, to figure out which are occurring more than expected by chance, and those are likely the important cancer genes. So where are we now? Where's the world of cancer gene discovery now? The quest to find all of the cancer genes that cause cancer is really reaching fruition. And I think in 5 years, we'll have a complete map of all the mutations for every type of cancer. That's really cool. Because how are we thinking about curing cancer if we didn't even have the map of what we were up against? Imagine sailing across the world without having a map. So we'll have the map. We have most of the map now. We'll have the majority of the map in 5 years. The first cancer genome was decoded in 2009. 6 years later, the Broad Institute, others around the world, tens of thousands, I think the number is about 20,000 now, of cancer genomes have been studied. And we built all these computational tools that are used around the world. I'm not mentioning it here, but there's a really exciting international coalition that's come together called the Global Alliance for Genomics and Health. This is a coalition of institutes around the world that have decided to share all of the genomic data that they're generating. If every institute did its own cancer genome sequencing, but no one shared it with each other, you would never be able to extract value from that information. So this is a moment in which the entire scientific community is coming together and saying, we want to share our genomic information, and this is needed, and the genome provides the rationale to do that. In addition to just studying samples in the lab, as I mentioned earlier, if you or a family member goes to Dana-Farber, MGH, with a cancer diagnosis, you will almost always now, in addition to someone looking at the cells under the microscope and saying they're aggressive or not aggressive, which is kind of the standard pathology, this type of DNA sequencing will be done. The set of mutations will, either looking at all of the genes or some of the genes, there will be a particular fingerprint. You get this report that says in your tumor, there are these particular mutations, and it's the hope that we would find drugs to give to patients specific for those mutations. And this is tremendously exciting, this rise of precision medicine. But there's 2 challenges, and I just have a couple, maybe 5 more slides. So if you're bored out of your mind, you have 5 slides. So there's 2 major challenges, and one of the challenges is the reason that we built this educational tool, which this biostatistics class has been piloting with us. It's actually challenge number 2. So challenge number 1, so we read off the list of genes that are mutated in Sally's tumor, and the doctor gets the report back, and the family gets the report back, and we say, okay, I know we have this roadmap, now we need to figure out what drugs to take. It turns out that only about 10% of all of the genes in the genome, drug companies have figured out thus far how to build drugs against. They call the rest undruggable. Everything's undruggable until you figure out how to do it. So they like saying it can't be done because that gives them the feeling that it's impossible. This is an area that we're trying to take on in partnerships with drug companies and partnerships with labs. I won't talk about that, but it's kind of cool to be working somewhere where every day people say, decide what's impossible, what other people say is impossible, and then go and make it possible. Eric Langer, the director of the Broad, has this notion, this way of looking at the world, which is really, I found very inspiring. He tries to encourage people to dream about what's needed. Flying cars or cancer drugs or whatever. Just write down what the world really needs. Put it over here. Now tell me all the reasons that can't be done. So it can't be done, can't have flying cars because you can't even build electric cars, you can't put wings on them, and you can't. And then he says, okay, you've identified the 17 reasons that thing can't be done. Now solve those 17 things. To me, that is a very hopeful way. It's kind of a parenthetical statement. You only get one chance to come to high school and say something provocative. I don't know if it'll be provocative or not, but to actually be able to conquer absolutely anything, as long as you can't just say it's impossible. You have to say why it's impossible. And if you can write down why it's impossible and devote your career to overcoming those reasons why it's impossible, whether it's going to Mars or putting devices in your body or making flying cars or, graduating from college, anything is possible. If you can specifically focus on the reasons it's not possible. Okay, that's total aside. 20% of the other genes, we know which ones to block, but drug companies tell us they're undruggable. You guys will help us figure out how to do that. But the second challenge was the reason we launched this project. So the other 70%, you have the genetic makeup, and your goal is to predict, given those genes in Sally's tumor, what drugs should she get? You have to really understand, there's 20,000 genes, there might in the future be 10,000 different ways of killing cancer cells. You need the dependency map. You need the way of reading off the genes and figuring out which way to kill cancer cells. So what we've been trying to do over the last several years, and now we're thinking about scaling this up dramatically, this is a big part of what my lab does and what the rest of the cancer program does, is to build a dependency map, all the vulnerabilities or Achilles heels of cancer cells in the lab. You can learn some of this information from patients, but that's very slow. So we want to do all the experiments in advance in the lab and build the map that then clinicians can read off. You also need people to read the map, and that's why we've enlisted CCHS students and others to help analyze the data. So I don't know, do people know about the course that you guys have been piloting? Is it worth?
To some degree, I guess.
As an aside, so
Tell the rest of them.
tell the rest of the French class may not know about so the pilot that we did was we built a matrix of genes, and I'll show you how we assess vulnerabilities or Achilles heels. And you need computational techniques to find the relationships that are statistically significant. You can imagine all sorts of random relationships that aren't real. And so we partnered with the Broads Education and Outreach Group and a series of high schools, including CCHS, which I was excited about, to try to make that raw data matrix, a real data matrix right off of the machines, make that available to students around the Boston area and then around the world, and try to encourage them to use existing and then maybe even in the future statistical tools to figure out the right relationships. And so the course was looking at a matrix, using statistics to figure out the relationships, and then trying to make a decision for the pretend cancer patients which had given you the particular genetic makeup. And so it's been a pilot, and you guys have been part of it, and you can tell us what's worked and what hasn't worked. But I really like this notion of trying to make data available to the world. There's been all these exciting, some 11-year-old in Bangladesh solves the amazing math problem, and you want to find all of the amazing expertise around the world that can help solve challenges. So to build these dependency maps in the lab, the first thing you have to do is grow cancers in the lab. This is actually, have people read this Gila book? Did people read this book about Henrietta Lacks and cells that grow forever and the exploitation of the family and all of that? Show of hands? Some people read
They
it.
have it in the library. It was one of the books that you guys got home.
Actually, I had to go, my wife teaches at Wellesley High School. The entire school had to read the book because it bridges politics and biology and law, and I don't know if that was a good idea or a bad idea. They all read it. So in any event, you can grow cancers in the lab. These are different types of cells that you can grow and what they look like. You can grow them from different parts of the body. And then you can figure out, well, you need two pieces of information to figure out how to kill these cells based on their genetics. One piece of information is, what's their genetics? So for this, I won't describe all the details. We've launched this big project. This happens to be a project in collaboration with Novartis, a drug company, but a company that wanted to partner with us to try to build a data set. They wanted it for some internal reasons, but we wanted to make it available to the world, but they were excited to fund that. So that's a new model of how to fund science. This is 1,000 cell lines. It's called the Cancer Cell Line Encyclopedia, representing all different types of cancers. All the different types of sequencing data has been generated. And so now what you can do now you have one part of the matrix. You have all the different cells growing in the lab and all the different genes that are turned on and turned off. The next thing you have to do is turn on and off each gene, test every single gene to see if it's a vulnerability in that cell. If you're a cancer patient, you can't take every drug on the planet and figure out which drug works. So you can only do a couple of clinical trials, perhaps, but in the lab, you can take a cell and you can block every gene one by one and figure out which genes make cancers die. And there have been a couple of technologies that are really important to know about. One that sort of set the stage that you may have learned about, maybe you've learned about both of them, called RNA interference. And the other that you absolutely have to know about because it's changing the world very, very rapidly. It's on like every front page, almost every New York Times. You're going to know about it in a year if you don't know about it now. It's called CRISPR. So in 2000, there used to be no real way of turning on and off genes in the lab. It seems kind of an easy thing to do, but there weren't drugs that helped you turn on and off the genes. There was no way of kind of cutting and pasting the genetic material itself. So scientists stumbled upon this way of interfering with RNA. Remember that there's DNA in the genome, and that gets turned into mRNA that makes the message, and the message gets turned into proteins. Did I get that right? So the message, that middle part, is RNA. And from a series of studies in plants and worms, scientists figured out how to silence that RNA. And so that has actually revolutionized turning on and off genes in human cells, but it turns out in retrospect it's not as specific a technique as we hoped for. And all that means is that only about 20% of the action is hitting the gene we think it's hitting. This new technology, just in the last 2 years, which is taking over the planet in an exciting way, is called CRISPR. This CRISPR technology on the left is a fancy animation that I'm trying to explain, just showing how it works. This is a tool originally found in bacteria. This is a type of genetic material originally found in bacteria. So everyone thought that bacteria had no immune system. How could bacteria have an immune system? It's just a cell. But it turns out that bacteria get barraged with all of these viruses that try to infect them all the time, called bacteriophages. And to be a bacteria that really makes it, you got to remember, you think evolutionarily, you want to remember which bacteriophages have hit you in the past so you can be ready the next time it hits you. It turns out that scientists had discovered that the bacteria actually remember which of these viruses they've already been infected with so that they're ready the next time. And they have this part of their genome called CRISPR, clustered, repeat, I forget the details. In any event, this series of memory in a bacteria cell is a way of using genetic material to cut DNA when they see it. So the bacteria then gets invaded with a new virus. It produces an enzyme that cuts the genetic material from that new virus. And in the last 2 years, scientists from the Broad and from Berkeley have figured out how to hijack this material from bacteria cells and use it in mammalian cells, like human cells and others. So you can actually put this whole system into a human cell and tell it to go to any place in the genome and cut it and turn off a gene. Or you could even tell it to cut the gene and then replace it with a gene that you want. So this CRISPR tool, specifically, you can go to one letter in the genome of 3 billion bases and make the change that you want. This is leading, there's been a series of studies, there's a lot of concern now about making designer embryos. You could, in the future, imagine a horror movie of turning on and off, parents creating the embryo that they wanted, and which could be for all sorts of bad reasons. If your family suffers from a genetic disease, you could think about whether it's ethically sound to make that change or not. But while the public press has been really focused on these doomsday scenarios of harnessing this to make genetic changes in embryos, scientists have been busy in the lab using this to turn on and off every gene, for instance, in cancer cells, and really carefully figure out what are the vulnerabilities. So you gotta follow CRISPR. So what we're doing, which led to this project, and this is the last science slide, is we're trying to build this dependency map. We think this is going to be the next frontier for the next 10 years. We already have the genome map. We already know all the genes. So if I come back, here's it, 2015. If I come back in 2025, hopefully, I don't know if Mr. Atlas will still be here. No, you're not going to be here. 2025. I'll tell you, we're done with the dependency map. On the left, we're turning off every gene one by one and figuring out if cancer cells survive or die. On the right, we have all of these cells growing in the lab and all of their genetic material, and we're trying to systematically find all of these relationships. When cancer cells die, what was the genetic change which made them die? Sometimes it's simple, like when that melanoma patient I showed you, they have the mutant gene, and you block the mutant gene and they die. But a lot of the time it's much more complex. That cell has that unusual combination of 14 genes, and they only die when you block another gene. So it's a big combinatorial problem, and you need statistical analyses to extract these relationships. And patients are helping to get us this information too. So if you read the New York Times, you've also seen these really exciting examples of cancer patients which thought that they weren't going to make it for very long because their doctor told them, with this type of disease, 95% of people, don't make it longer than a certain amount of time. But certain patients, even for drugs that fail, so a drug company might develop a drug, they test it in 100 patients, it might only work in one patient. Amazingly well if it works in one patient. The drug company abandons the drug because they wanted it to work in every patient. Gets shelved put on the shelf, it's a biomedical mystery. What's this woman's name? Why did Grace respond to that drug? And until you had the human genome or the cancer genome to study, you could never figure out why Grace's tumor responded. But now you can use genetic sequencing to go back and say, why did Grace respond to the drug? And now if you know why she responded to the drug, now you can only give that failed drug now to patients just like Grace. So these are called exceptional responders. These are patients that thought they weren't going to make it and are now alive many, many years later to tell us its tale. And there's an international effort. The National Cancer Institute is leading the way. We're doing some stuff to find these patients. This social media project I was talking to you about, that is designed to try to go out there and find patients that have had unusually profound responses to therapy. It turns out that if you get a drug and you are still alive 10 years later and someone told you that you're only going to be alive for 4 months, you tell a lot of your friends and you form a community. And so we want to try to find all of those people across the world and invite them to be part of the study. So the final thing I'll say, and this is just more sort of waxing and waning, I think, about this community aspect. Some of you may go on to be scientists, some of you may not. Obviously, if it was up to me, you'd all be scientists. That's just me. But it's very important to realize, and I've tried to make it clear in a few places in the talk, and I'm trying to make it clear in this slide, that everyone is really today part of this scientific community. Whether or not you're a scientist, it doesn't matter. It's not an excuse to not understand science. If you go to the doctor, take medicine, vote, use social media to look up disease information, you're part of the scientific ecosystem. You're making decisions about should we put more money into solving disease or less money? If I respond to a drug, should I make that known or not make that known? Patients, as I told you, may hold the keys within their own genome to solving diseases. I didn't have time to tell you about rare diseases, patients with really unusual symptoms that go on for 10, 20 years never knowing why they had those symptoms. But now with genetic information, genome sequencing, you can figure out why those patients have that collection of symptoms. And if you can find the 7 patients on the planet that are similar because of that genetic information, they can come together and form a community. That's actually very empowering. So patients actually hold much more of the fire to solving disease, even than scientists today. And we're all patients of one thing or the other. Just like the project that we did with you guys, these crowdsourcing challenges to send data out there and ask people for help in solving problems, that's an example of, I think, a trend to really lower the barriers to engage with science, even if you don't have a PhD. And as I showed you on that slide with the different grids, you can propel science even without being a scientist. Policymaking, law, counseling, genetic counseling, and relation building to give advice about genetics and decision making. That's a really big growth area and a really exciting career. You don't need to do 30 years in biomedicine to help give advice to patients. Advocacy, communication and marketing, social media, technology, and Google and Apple. You probably got your Apple. You guys have iPhones? Does everyone have iPhones now? Okay. Who has an iPhone? Okay, everyone has an iPhone. Okay, so there's like the health kit thing now that just appeared on your home screen. Apple is really getting into this business. Google is as well. Apple Watch, you can monitor your steps and everything. There's a Parkinson's, there's a set of apps that are connected to HealthKit that scientists are using to try to find research participants. There's a Parkinson's app which monitors your steps and your movements, and you say that you want to be part of the Parkinson's study, and then it helps you to decide whether your movements suggest that you might have Parkinson's. So using technology and social media and big data collection in Silicon Valley is like totally the next generation. And so there's this interesting relationship. Where's the future of biomedicine going to be? Is it going to be driven by Silicon Valley and startup companies, or is it going to be driven by like the Boston area, which is the home of biomedicine? I hope it's Boston, but I think careers in both of those places can have an important impact on science. So that's it. So it's 40 minutes. That's okay. So I tried to take you through a bunch of stuff. I tried to think about what would have made me excited. Maybe it did make you excited, but if you're not excited, at least realize that I'm excited. And the reason I'm excited is because I think I can see where the field is and where it could be going, and I'm part of a community that feels similarly, and I feel very fortunate to be part of this community. This is also parenthetical, I'm making up a totally new career path that just doesn't exist. I don't know if that's okay to say. Most people in my situation go to grad school, get their PhD, start a lab, put walls around their lab, and compete with everyone else around them. That's the way that we reward science. You write your grants. If you share data with your neighbor, they might write the grant first and get them. So the incentive structures of the world promote competition. I decided I didn't want to be part of that. I wanted to build teams and solve problems that were beyond what me and my group could do. And the Broad is a place where the culture of science is, I think, changing. And so I'm trying to build a new type of research effort that is actually enabling others and not just myself. And I don't know if it'll succeed or not, I'll let you know, but even being limited by what career paths exist, I think is limiting. You guys will make up new career paths that don't exist that might be in between these different areas, and that's okay too. And then someone will say, oh yeah, you can do that, so I'll do it. So I've tried to take you through science and waxing and waning about biomedicine. Maybe questions? Question about iPhones, I can maybe answer questions about genetics, question about French. I definitely cannot answer because it's Spanish.
Just really quickly, you mentioned growing your cells in the lab and turning on and off every gene. How long does that take to do that?
So the cells, you would think that cells that grow in your body can grow really rapidly in the lab. That turns out not to always be the case. It's usually about a month to grow in the lab, and then maybe another month to